Okay, today we're doing a pre-lab, and this is a pre-lab on mitosis. Mitosis is a process by which we produce every cell in the body. So originally, of course, to create an animal, then the female produces the egg and the male produces the sperm. Well, in the case of a human being, the egg is going to have 23 chromosomes, number 1, number 2, number 3, so forth. The sperm also carries 23 chromosomes, which would be number 1, number 2, number 3, and so forth. So when there is a fusion of the sperm and the egg, then you actually get 2 number 1s, 2 number 2s, 2 number 3s, 2 number 4s, and so forth. So we actually get 2 copies of each chromosome, which that's sort of a backup system in case one chromosome becomes non-functional for whatever reason. Then you actually get actually 2, and then so you have one that is still functional, which is very beneficial. Okay, so anyway, once we get the total of 46, 2 1s, 2 2s, 2 3s, anyway, 2 number 23s, which of course all together we would get 46 chromosomes, and that would be the entire genome of the organism. Then that cell begins to divide repeatedly from 1 cell to 2 cells to 4 cells to 8 cells until we end up at whatever stage of life we're at. And so we're at about 70 trillion cells at this point. Well, in order to get to that point, we've just had multiple cell divisions, and that whole process by which you take 1 cell divided into 2, those 2 cells divide again into 4, and so forth. Those are mitotic cell divisions. So we're actually doing a mitosis, and so the cells that we're producing are actually identical to the original cell, and so it's what they sometimes refer to as quantitative and qualitative cell division or nuclear division. Because what you're doing is you're taking 1 cell that has 46 chromosomes and you're actually producing 2 cells that have 46 chromosomes. So the quantity, the number of chromosomes is the same. The quality is also the same, meaning that the 46 chromosomes that we have produced are exactly identical to the 46 chromosomes that were in the original cell. So therefore it's quantitative and qualitative, and it is a nuclear division. Okay, so if you'll notice that what we have here is we have really what is referred to as the cell cycle. The cell cycle is divided up into different phases, and so originally what we will begin with here, so we'll just put the cell cycle, and what we'll start with here will be what we refer to as an interphase. And they call this interphase because originally they didn't think much happening during this period of time. So we further divide that. They have since come to learn that there's quite a bit of activity happening here. And so we have what is called a G1, and that is really means gap 1. And that, of course, is just saying that there's not a whole lot happening. But in the G1 phase, that is where we're actually having quite a bit of growth of the cell. So this would be, say, for example, if we had 1 cell and it divided into 2 cells, what you get are 2 cells that are 1/2 the size of the original. Well, what has to happen with each one, they have to grow to the size of the original cell, and so each one will do that, and that's what is referred to as the growth phase of interphase. Be what is the S phase, and the S phase is the period during which the chromosomes will replicate. So if I have, let's just say, this 1 chromosome, when you're finished replicating it, you actually have 2 copies of that chromosome. So there is 1 and here is a second, and they're exactly identical to one another. You can see that they're both held together. That area where they're held is called a centromere. So it holds the 2 identical copies of the genetic material together. Each copy that we have is referred to, instead of calling that a chromosome, the identical copies are referred to as sister chromatids. Okay, and the entire set of both sister chromatids, that really would be the entire set, not, well, actually both sister chromatids together. That is actually one chromosome. So we have actually two identical DNA molecules, but as long as they're held together, that would be one single chromosome at that point. Okay, and then we have the phase of the cell cycle, which is referred to as G2, and that does stand for the gap phase, but it's a period of tremendous amount of synthesis. So we have to produce a tremendous number of proteins in order to divide the cell. In order to do that, many of those are enzymes, many of those are structural proteins. Say it's a period of protein synthesis. Okay, the time frame in order to do this entire cell cycle, somewhere in the neighborhood of about 19 hours. So this is a very long period of time. And if you're looking at a cell going through interphase, what you will see basically is you see a plasma membrane on the cell. You see a nuclear envelope, that is a double membrane structure. And you really don't even see the chromosomes inside. You may see a dark stained structure, which is referred to as a nucleolus. And that's where the ribosomes are produced. The ribosomes, of course, are going to be where the proteins are manufactured. That's where protein synthesis occurs. Okay, so what you do see, though, is you kind of see just a meshwork of material in here, and that's really all of the 46 chromosomes, but you can't really make out any one chromosome. They're kind of in a loose, uncoiled state, and all of those collectively, which would be all 46 of those, are loose and have not compacted to the point that they're visible. So in that state, they're referred to as chromatin, the entire 46 chromosomes that are all in a loose, uncoiled state. All right, so that's basically what you would see under the microscope. Again, this is all what we refer to as the interphase of the cell cycle. Okay, so the next phase that we end up getting, what is referred to as the mitotic phase. So that is actually mitosis, mitotic phase. Sometimes they just refer to it as M phase, and sometimes, of course, this is referred to as mitosis. Trace a little bit of that. We have gone to black. All right, back to blue. So mitosis, or mitotic phase, or M phase. All right, now that is divided up into what we refer to as prophase. This is the chromosomes condense, they become visible, and the nuclear envelope disappears, and then we get into what is now referred to as prometaphase. This is something that is introduced, I would say, within the last 5 to 10 years, and that is a period during which the condensed chromosomes, which at this point they should be visible, actually attach to what we refer to as spindle fibers, which will pull the sister chromatids apart. So that's when they actually do the attachment to the spindles. And then we go into a metaphase, and the chromosomes line up in the center of the cell, and they call that the metaphase plate. Sometimes they call that the equator. Depending on the book, there are different terms for that. and then we get into what's referred to as anaphase, and that's the period during which the sister chromatids are going to be pulled apart, and so half of those will go to one side of the cell and the other half will go to the other side, and then we get into telophase, and that's where the chromosomes have reached the poles. So we actually have 20, well actually we have 46 on one side of the cell, 46 on the other side of the cell, and then we do have a reformation of the nuclear envelope around both sets. The last phase, and this is all seen in the in the lab book, but the last phase we have, which will be the third phase, is the cytokinesis. Cyto refers to cytoplasm. Kinesis actually means, it's a Latin word, which means cell division. So this is the period during which the cell actually is going to begin to pinch into 2 units. So that will, of course, continue until you have divided that cell into two cells, one half the size of the. Now, this would be for the animal, and then for the plant, what happens? You can't really pinch it, which is to create what they would refer to as an animal furrow. And there are proteins that attach to the plasma and draw it inward. As you can see here, it's beginning to pinch into 2 units. So proteins attach and pull them in and sort of contract it at the center of the cell. Okay, now you can't really do that on a plant. So with a plant cell, because it has a rigid cell wall, so it's more of a boxy cell, and the outermost part of that, of course, is cell wall, and it's a very rigid structure, so it won't actually pinch. So what happens instead, and I'll draw this a little bit larger, begins to happen the Golgi status. So we'll have chromosomes at one end of the cell and reform a nuclear envelope, and chromosomes at the end of the cell and you reform a nuclear envelope. But you have the Golgi apparatus, and the Golgi apparatus is what produces our little vesicles. Well, those tend to be produced in large numbers, and they line up through the center of the cell, and this is referred to as this entire plane that they've created here of vesicles. That is referred to as the cell plate. And for some reason, a lot of people confuse it with the cell wall, which, of course, the cell wall is the outermost portion of the cell. So that would and that's vesicles that which happens frequently. Okay, those vesicles will eventually fuse, and then at that point you really have created a cell that is divided into 2 units. You've created a division between the 2. So you do create a cell plate at that point. Okay, so we'll look at this, and we'll look at slides that do demonstrate this, and we'll look at some models that demonstrate this, and of course you'll have some diagrams that will also help us to see this. And then also you'll see some diagrams and photos of animal cell division as well as the plant cell division. Okay, so that's a pretty short little preview, and I guess that will be all for this preview, our pre-lab, and